Chapter 16, Adaptive Immunity. This is the third line of defense in the immune system. And this line of defense heavily involves the activity of the lymphocytes. And I see a typo right here. And these are the study objectives. So, just like in the last chapter, maybe making a chart and helping to lay out what each cell does, and a lot of cells have multiple roles and they interact with each other. It can get pretty complicated. We're trying to look at this at an introductory level, and so I will try to simplify it as much as I can. And then if you take an entire course on immunology, you'll see that there are more interactions and such. But basically, if we can define and know what the cells are that are involved, some of their basic interactions, and there's a list of vocabulary that is a big piece of being able to understand what's being presented. So we're going to go through these basics. There are several really good animations provided by your textbook that I have uploaded into eCampus for you. I strongly suggest you watch those because they would have been presented during the on-campus lecture, and it really helps to put the different interactions together visually. All right, let me go ahead and get started. Adaptive immunity is the third line of defense. This is the line of defense that is mounted against a specific pathogen. So every different pathogen that comes in would get an adaptive immune response, but it would be tailored, it would be custom made to that particular pathogen. So this is where antibodies are produced, which are specific to particular pathogens. Not even just the general pathogen, but like the exact pathogen, especially with viruses that mutate every year. You will have an adaptive immune response to the flu. Let's say if you get it this year, next year when it's mutated, some of the antibodies that you have for the flu would probably still work, and then some new ones would have to be made in response to the changes in the flu after it has mutated. So, but this is the third line of defense. So this is an example. This is one of the videos that I have posted for you. This might be a good time to stop this video and then go to eCampus and watch the overview of adaptive immunity. So what you'll see in that overview is there's really two responses. If we're going to start at the very basics, in adaptive immunity, in the third line of defense, you have what's called the humoral response, or the antibody-mediated response. It's called humoral. The humoral response and the cell-mediated response. Now, there is some interaction between them, but we try to kind of address each of them separately as much as we can. And fundamentally, you have two types of lymphocytes that are working. The B lymphocytes, the B cells, these are cells, all the blood cells form in the bone marrow from stem cells, blood stem cells. And the lymphocytes, there's one group of them that stay in the bone marrow, and that's where they mature and they go through a process called clonal deletion, which we'll discuss later. And they stay in the bone marrow during all of this, and then they are released out into the blood, clonal deletion. T cells, T lymphocytes, form in the blood, in the bone marrow from stem cells, but then they move to the thymus, and that's where they mature, and that's where they undergo their clonal deletion. So the name of the lymphocyte, the B stands for bone marrow, the T stands for thymus. That's where they mature. They're both formed in the bone marrow from blood stem cells. The B cells stay in the bone marrow, the T cells move out to the thymus for their maturing and their clonal deletion processes. Blood stem cells is what I was trying to write. All right, so roughly speaking, the B cells are mostly involved. I'm going to connect these two. The B cells are mostly connected to the antibody immune response, the humoral response, and the T cells, roughly speaking, are more involved with the cell-mediated response. So the term cell-mediated isn't maybe that helpful because all of these things are cells. But the cell-mediated response is not particularly the release of antibodies. The antibodies are released from the B cells. The cell-mediated response is some other parts of the adaptive immunity. But roughly speaking, we link the B cells to the humoral response and the T cells to the cell-mediated response. So we need to talk about how the body knows, how those B cells and T cells know when they should respond. Because you don't want to have an immune response if there's no pathogen. Because remember, the immune response, you're going to have a lot of activity, you're going to put a lot of energy into this, and if there's no pathogen, that would be a waste. So how does the body know when something is there that should not be there? There are on all cells antigens. And this is just, all cells have antigens. Usually antigens are macromolecules, usually proteins or glycoproteins on the surface. So let's say proteins or glycoproteins is usually what we're talking about. And you'll remember glycoproteins are proteins that have some carbohydrates attached. So the protein concept is very important here in terms of what is an antigen. And they have to be on the cell surface. So they have to be either transmembrane, across the membrane. They can be peripheral on the outer surface, maybe. So but usually like a transmembrane protein is typically how these things are depicted. So here's a transmembrane protein. I don't know if you remember the phospholipid bilayer here. So they're showing you a close-up of the cell membrane. You're zooming in. This is just any generic cell. On the surface, on the membrane, you'll have part of the transmembrane protein that sticks to the outside and part that sticks over onto the cytoplasm side. But remember that proteins are have three-dimensional shapes. They have these three-dimensional shapes. And so different parts of these three-dimensional shapes, here they show, this is kind of ridiculous, here they show like a cone, half of a sphere, and some kind of weird thing. They would not really look like this. They're more irregular than this. But you have these different shapes, so they're representing those here. Each little section of the glycoprotein or the protein can be what we call an epitope or an antigenic determinant. I will typically use the term epitope. That's how I was trained. An epitope is really just a smaller section, a small subsection of the larger antigen, which the antigen is really just a transmembrane protein. It can be a foreign cell, it can be on your own cells. Now your body though, your body can tell the difference between your own antigens we call self, that's simple, And anything that's on a foreign cell, a pathogen, or just any cell that's not part of your body itself is called foreign or non-self. I tend to use the term non-self because that's how I was trained, but those mean the same thing. So, and it's also true that the larger the protein, the more epitopes it will have, and the more immune response you would get from it if it's a non-self antigen. So epitopes are just regions that potentially can be recognized by the immune system. Your body, though, through a process called clonal deletion, will eliminate any B cells or T cells that react to self antigens. So if you have a B cell or T cell that would react to your own antigens, the body gets rid of those B cells or T cells so that you don't have a self-reaction. And what that leaves are any B cells or T cells that are left after clonal deletion are the ones that would react only to something foreign, something non-self. That way you prevent autoimmune disease. So most of us do not have autoimmune disease. That means for most of us, the clonal deletion works properly, and all of the what we call self-reactive or sometimes called autoreactive B cells and T cells are eliminated very early, like when you're a baby. If you do have an autoimmune disease, that's some kind of malfunction of clonal deletion, probably. It's probably what it is. So there are malfunctions of clonal deletion, sure, but most of us do not have an autoimmune disease. That means that this process works correctly. There are transmembrane proteins, glycoproteins, that all of your cells have, and they're called the major histocompatibility complex proteins. These are the same on every cell, and you have two types of them. You have one type that's called class 1. And the MHC class 1 proteins you have on every single cell in your body. The only exception to that is red blood cells. Red blood cells, they don't have a nucleus, and they also don't have class 1 MHC, which makes them kind of special. But all the other cells have MHC class 1 transmembrane glycoproteins. The MHC class 2 transmembrane glycoproteins are only present on antigen presenting cells, and we're going to define that. And antigen presenting cells are, for example, your phagocytes. So you probably remember this is where some of the second line of defense sort of crosses over and has some function in the third line. So macrophage, you'll remember, is what we call a monocyte when it enters the tissues. Dendritic cells, those are in the dermis of the skin. You have some other phagocytes. So the antigen presenting cells, you can think of them as being the phagocytes. So not only do they perform phagocytosis, which is part of a non-specific killing initially, they then, when the pathogen is broken up inside that phagocytic cell, they then stick parts of that pathogen on their cell surface. So here's your pathogen. Now that's too big. Here's your pathogen. It gets engulfed by a bigger cell, that's your phagocyte, so maybe a macrophage. And then this gets all broken up, and then the macrophage puts little bits of it on its surface, little pieces. It's really kind of grotesque if you think about it, because not only does it kill the pathogen, then it takes parts of the pathogen and sticks it on its cell surface and dances around. Well, I say dances around. That's how I imagine it. Like some kind of crazy psychopath advertising what he has just killed. So it's kind of dark. But that's what we call antigen presentation, because the little bits of the pathogen are now being displayed, sometimes we call it display, on the surface of the macrophage. And it could be a dendritic cell, it could be any kind of phagocyte. So that's called antigen presentation, because the cell is showing the other cells in the immune system, hey, look what I just destroyed. Those antigens from the pathogen are presented with what we call an MHC class II molecule. So they're presented together. So you have the MHC class II, and then you have some bit of a pathogen attached to it on the surface of your phagocyte. And that's called antigen presentation. Here is a picture, a drawing. Here's your phospholipid bilayer. Let's say this is your, well, in this picture they combine two things. If the cell is just a regular cell, a regular body cell, it will just have what we call the class I MHC. So it's a transmembrane protein, and it has a little place here where an antigen can be attached. It doesn't have to have an antigen, but it might. If this is an antigen presenting cell, or I didn't mention B cell, then your MHC class II also has the transmembrane protein and a binding site. The structure of it's a little different. You can tell this one only has one transmembrane section, this one has two. The actual protein is different. But what I'm saying is they both have a place where an antigen can be attached. So an antigen presenting cell will always attach, a piece of a pathogen that it just took in, it just digested. It will always stick it into an MHC class II molecule and put it out on the surface. Typically what MHC class I would be for is little pieces of proteins that are normal parts of you, self, would be sometimes presented here or just in the membrane. So the only other exception though is MHC class I will also display virus bits. So virus bits, if the cell is virally infected, then it will just present some pieces of virus antigens here. So self or virus will be presented with class I MHC. Class II MHC is restricted, only certain cells have those, and particularly you want to think about your antigen presenting cells like your phagocytes. So this would be the bit of a bacterium that cell just digested. And it's displaying it out. All right, it's displaying that antigen. All right, so here's an example of how that looks like. So here's your phagocyte, actually much better drawing than what I did. Here's your pathogen. So here's your pathogen here. Now what they're showing here in green are these little antigens or maybe epitopes of antigens, whatever. The cell takes in the pathogen, and remember, it makes a food vacuole. The food vacuole fuses with a lysosome. There's digestion. And then after that part is done, you get to this stage. So here's the food vacuole, sometimes what we call the secondary lysosome. After the digestion has occurred, then what will happen here is these little bits of the pathogen, when this vesicle fuses with this vesicle, this vesicle has MHC class II already in the membrane and they're facing inward. When these two vesicles fuse, you see here over here, it will load those bits onto the MHC class II molecule binding sites. And then this whole thing goes out to the cell surface and fuses. And you can see when this sort of straightens out, then it will result in the presentation of those pathogen epitopes. So this is what is a processing and a presentation of antigen. Now we call them exogenous antigens because it was something that wasn't in the cell initially. It was brought in and processed and then displayed. An endogenous antigen would be something the cell makes, like self. Self is an endogenous antigen. So any proteins that are part of your body normally would be called endogenous antigens, normal, self. The other thing that can be an endogenous antigen, though, is a viral antigen because it's inside the cell when it's causing trouble. So when a cell displays an endogenous antigen that is a virus piece, then that will trigger the immune system. But this one, MHC2, displays exogenous antigens, which you can think of as being antigens on a bacterial cell that was phagocytized. So I'm going to stop here and give you a chance to watch two videos. This one's called Antigen Presenting Cells, and the next one looks the same, but it's not quite the same. It has to do focusing specifically on those major histocompatibility complex proteins for presentation. So there are some differences here in these two presentations. They start sort of similarly, and they focus on different aspects of this process.